Okay, so this is Mock Viva, lecture number 2, which is on gastrulation, primary germ layers, origin and fate of notochord, the neurulation stage, somites, embryogenesis, gray and white matter in the spinal curve. The different divisions are mixal spinal nerve, so dorsal, ventral roots, dorsal root ganglia, dorsal and ventral rami. So let's begin. So let's begin looking at this diagram here. So this diagram is basically of the uterus. And what happens in the uterus is the uterus is involved in contraction, which actually leads to pregnancy. The increase in the calcium or the potassium ions actually lead to further stimulation in the electrical stimulation. So you have something called polarization here. So polarization is actually different to chemistry here. The reason why it's called polarization is because one end is more positive than the other one. So let's have a look at this end. So at this end, we have the more positive end here and the more negative end here. So what basically happening is we're going to stimulate that push of charge. So it's going to become positive. And this actually results in further stimulation. As you can see, the different blastocysts. So the blastocyst is actually formed of various different layers. So it forms of many different hard words that I always fail to pronounce. Forms the chopper class and synchro class or something like that. And then we have the morella stage. So what basically happened here is the morella means a bunch, and it's basically like a blueberry shape. So what basically happens is you have the morella, and this stage actually forms the is actually the basis of the zygote stage. And what basically happens is you have cell division. So you have 8 cell, 4 cell, and 2 cell. And the reason why it decreases is because you want that mitosis and meiosis replication. And that's something that you're going to do. Now let's further differentiate the difference between mitosis and meiosis. So mitosis has two different stages, and it's mainly split from one daughter cell into one nuclear cell into two daughter cells, whereas the meiosis stage occurs twice, and there's two meiosis stages, meiosis 1 and meiosis 2. But you don't need to know that further depth at this stage because it's not really relevant to anatomy, more developmental biology. So let's moving on. So the next thing is we have the two-stage cell, and what happens is we have a zygote. And what basically happens during the zygote stage is you have an oocyte. So you have an oocyte and a sperm. And what basically happens is the sperm, it consists of an acrosomal head, mitochondria, and body. And what basically happens is the mitochondria basically drives the sperm into the oocyte. And what basically happens is it cuts its tail off. Very grim, I know. But the reason why it does that is because it needs that nucleus to propel itself into the body, into the fallopian tube. And that's essentially what's happening here. And the only reason why is the zygote only needs one sperm. So what basically happens is we have basically a membrane, and it's actually very interesting because it basically it uses calcium charges to basically prevent other sperm from binding to the membrane. I forget what the term is actually called the pellucida. Um, yeah, so that's the membrane. So then what essentially happens is it moves into the fallopian tubes and it gets broken down, as you can see. And it forms different maturing follicles before it's released. And that's basically how pregnancy occurs. The epithelium consists of many different layers. You don't really need to know that for now, but you need to know it for the respiratory series. But that's about it for this stage here. Next, we have gastrulation. So gastrulation is the formation of three different layers. We have the we have the endoderm, which is mainly your digestive system and internal organs. We have your mesoderm, which is your circulatory system and the stuff in the middle. And we have the ectoderm, which is mainly involved in the brain, spinal cord, and skin. And as you can see here, we have the dorsal and caudal, the cranial, and the ventral end. And what basically happens is the way that gastrulation forms is it has an epiblast and a hyperblast. And what basically happens is it basically migrates into the primitive streak and it forms a groove called a primitive groove, which forms the epiblast and the hyperblast. The hyperblast, from my understanding, forms the mesoderm, I think. And the epiblast actually forms the ectoderm and the endoderm, I think. But we'll double check that later. So these are the different views. Yeah, as you can see, this is the neurulation stage here. So what basically happens is you have two ends here. You have two ends here, which is a neural fold, and the neural crest, which is actually involved in the outer regions of the sections here. And what basically happens is invagination folds, and the nodal cord can actually push through the push through the migration. And so what basically happens is as it's moving forwards to each other, as it's moving forwards it pushes forward and it pushes the neural crest outwards into the ectoderm. And this actually forms the ectoderm layer here. So what essentially happening is we're forming the ectoderm and the neural tube. And what happens is there's two different ways that it can form. So there's the rostral and the dorsal nerve form. But if it doesn't correctly fold, can result in spina bifida, which is basically where the neurophore or the neurulation tube does not form properly, and the neural crest is actually located in the inside the neural canal. So canal means hole, and this is the tube. Next, we'll move on to the analysis here. So we'll move on to the dorsal root and the dorsal root ganglia ventricle root. So the dorsal and ventricle root, one is involved in the motor. So this is, you have the motor here, and you have the dorsal root and the dorsal root ganglion here. And here is the formation of the neural tube. Yeah, this is further analysis and different view of the dorsal horn, the ventrotron, and the intermedial lateral. So intermedial lateral here means inter, so that's inner, and the medial, which is the middle, but it's towards the lateral end. So it's going to be further away. Hence the word intermedial lateral. Next, we have the formation and the different layers here of the vertebral crest. So we have the lateral mesoderm here, which forms on the outside, and we have the somites. So somites are basically involved with the mesoderm. And yeah, that's our full analysis of this one. I know that I didn't cover all of them, but I covered the main different parts that were related to Lecture 2 gastrulation. And that's basically my analysis for the Viva Lecture 2 on the gastrulation, germ layers, origin and fate of the nodal cord. So note that the nodal cord actually forms the brain, and the neurulation stage, somites, embryogenesis. There's also stuff like spermatome, myotome. So myotome forms the muscle, spermatome forms the dermatome and um, another term, but it eventually forms the epimere and the hypermere, which actually relates to later, which is the apaxal and the hypaxal muscles, which I think was covered in this lecture, but it's not covered here. So we'll just leave it for now. And that's basically my analysis of Viva 2 Lecture 2.